Welcome to Lecture 5 of our Hereditary Genetics unit. We're going to talk about linked genes to start with. So a linked gene is when genes are on the same chromosome. So Mendel's law of independent assortment can be applied when genes are on different chromosomes, but not when those genes are on the same chromosome. So let's take a look here. Here we've got genes A and B. They are on different chromosomes, okay? Versus here, genes A and B, you can see, are on the same chromosome, so they are linked. So when you complete meiosis, over here on the normal gene, A and B are going to be on separate chromosomes. Over here, they will remain on the same. So before we get too much further, let's do a quick review. So 26% of the offspring shows recombinant phenotypes for a gene for eye color and a gene for fur color. Calculate the distance between the gene for eye color and the gene for fur color in map units. And remember our rule, the distance between genes equals the percent of the recombination or crossover. So our answer here is 26 map units, since they gave us this 26% here. Next one, the following genes are linked and show the crossover frequencies below indicate the order of the genes on the chromosome. So we've got A and C, there's 10% between them. A and B, there's 30, and C and B, there's 20. So we know that the biggest space is going to be between A and B. So if we draw our little chromosome here, let's say we put A here and we put B here. And we know that that's going to be 30%, or 30 map units. So we have to figure out, then we have C, which is 10% from A, or 10 map units, and 20 from B. So that means that if we put it here and we label this as 10 and that's 20, then it works out. So your answer would be ACB. Or you could do it BCA also. Okay, so let's move on. So we need to be able to determine if genes are linked in a dihybrid cross. So we've only looked at dihybrid crosses with genes that are unlinked. So if a dihybrid cross shows unexpected offspring frequencies, you need to consider that the genes could be linked. So there's a big clue. If you have a dihybrid cross and there are two phenotypes and they have similar frequencies, and the other two phenotypes have a similar frequency, then the genes are likely linked. So let's take a look at what we mean. For instance, here we have a cross between wild-type eyes, wild-type body, mahogany eyes, and an ebony body. You can see that right here, there's eyes and there's body type, and these numbers are very similar. Right here, the 370 and the 380 and the 130 and the 120. So these are likely linked genes. This here, we have body color and we have development of wings. And again, they're very close in numbers, so these are probably linked genes. So let's look at the history a little bit here of the linked genes. So Thomas Morgan was one of the first true geneticists, and he studied fruit flies and their traits, and he crossed them. And he contributed greatly to our general knowledge of chromosomes as well as the inheritance of genes. So what he discovered was that chromosomes can carry multiple genes, and indicated that the genes on the same chromosome are linked and would not result in data to other dihybrid crosses with traits on different chromosomes. He also hypothesized about crossing over. Now, this was in the early 1900s, so this here was his illustration of crossing over. So his famous experiment was he crossed a heterozygous female that were gray with normal wings and males that were black with vestigial wings. And he expected that he would get one gray normal, one gray vestigial, one black normal, and one black vestigial. So if we do that cross, and remember, we're going to do a short one because of this GGNN right here. So we know that this right here is GN. And then we've got the other combinations. Capital G, capital N, capital G, little n, little g, capital N, and then little g, little n. So if we put all those in, we should end up with this information. Okay, so basically what he ended up getting, or what he observed, is that, or let's back up 1 second. So what he expected, basically, is a 1-to-1-to-1 ratio. So let's say that he got about 203 here, 199 here, 205 here, and 198 here. So that's pretty close to a 1-to-1-to-1-to-1 ratio. What he actually observed was 203 gray normal, 195 black vestigial, 4 normal black, and 6 gray vestigial. So when observed results do not fit the expected, geneticists have to determine why. So in this situation, the two genes for body color and wing shape were actually linked. And these fruit flies resulted due to crossing over. So these organisms are the recombinants right here, the 4 and the 6. So this is basically what we're looking at that occurred during meiosis. So basically you ended up with a crossover right here, and that gave us, let me see here, get my pen ready. If we do that cross, we still have our big G, our big N, little g, little n, but then crossover occurred, giving us a big G, little n, and a little g, big N. So because of that crossing over, it changes what we end up with. Since this only occurred in a few spots, that's why there's only 4 or 6 in this area versus the expected number. So if we go ahead and do this, So right here, we ended up with the 203, the 195, and then we have the 4, and we have the 6. Okay, so let's look at some linked genes related problems. If genes are linked, you could be asked to calculate the distance between those two genes. And to solve, you have to remember that the distance between two genes is equal to crossover or the recombination percent. So let's look at number 1 here. Morgan observed the following results. Calculate the distance in map units between the gene for body color and for wing shape. So the first thing that we know by looking at these numbers is that we have linked genes and that crossover occurred. So to determine this, we're going to take the the recombinants over the total offspring. So our total, if we calculate this, was 408. Our recombinants are these right here, which is 10. So we're going to take 10 divided by the 408. And of course, you want to multiply it by 100 to get a percent. And that would be 2.5%, or 2.5 map units. Okay, example 2. In a dihybrid cross, it was determined that one recombinant phenotype occurred 5% of the time. Calculate the map distance between the two genes setting in the cross. Okay, so remember we have to equal 100%. So if this phenotype is 5% and it's a recombinant, we know that there's two recombinants. So we're going to put each of those at 5%. So that leaves us 90 for the other two. So we're just going to divide those phenotypes, 45 and 45. Okay, so phenotype 1, 45, phenotype 2, 45, recombinant 1, 5, recombinant 2, 5. So our recombinant percent is going to be 10. So our distance between the linked genes is going to be 10 map units. So the reason why we do that, if you look up above at this first example, these are very similar in numbers. These are very similar in numbers. So since we only have this 5%, we are going to make some assumptions. Okay, so example 3. So this goes back to what we first talked about. We're going to have an expected ratio of 1 to 1 to 1 to 1. Remember that. So snapdragons with genotypes CCDD and little CCDD are crossed. The progeny were then crossed with the lowercase CCDD plants, and we obtained 30% of this, 20% of this, 20% of this, and 30% of this. You can conclude that. So this is where we're going to look at, are they linked or not? So the first thing that we need to determine is which ones are the recombinants. So since these are the lower percentage, these are going to be our recombinants. So looking, if we add these two together, that's 40. Remember, you have to add them together, so it wouldn't just be the 20. So our answer is going to be C. So most offspring of a cross with linked genes have a combination of traits that is identical to that of either parent or the other. The other offspring are going to be the recombinant. Okay, let's go to example 4. Two genes are 18 map units apart. What percent of the offspring would show the recombinant phenotype black body? Okay, so two genes. We're assuming that they are completely talking about the recombinant since they're asking about that. They're 18 map units apart. So remember, that's the only data that we have. We know that there's two recombinants. So we are going to take that 18 and we're going to divide it up. So we have a 9% and a 9%. So your answer would be 9%. Example 5. In some flowers, a gene for the length of style is found on the same chromosome as a gene for the color of the stigma. The style length is either L for long or little l for short. The stigma colors alleles are big G, little g, giving green and red stigmas. The diagram below shows the parental chromosomes. Okay, so here we are. So write the genotype of the two parents. Well, looking here, you've got parent 1 and you've got parent 2. So your answer is going to be LL and GG. And your other parent will be LLGG. If the if the genes were on different chromosomes and independently sorted in meiosis, how many different, there's a typo right there, my apologies, different gametes would the first parent produce? Okay. So what we're going to do is we're going to do our crossover, just like we normally would. So we are going to cross this L with the big G, L with the little g, okay? So first parents, big L, big G, big L, little g, little L, big G, and little L, little g. So that gives us 4. Show a cross to determine the genotypic ratio of the offspring. Assume the trait is linked and no crossing over would occur. Okay, so now we are going to have this crossover between parent 1 and parent 2. So we're expecting to get a 1-to-1 ratio. So the first thing that we have to determine is what we're going to cross. So we've got an LG and a little LG. Right? Parent 1, right here. And here we know that we're just going to have LG. So we're going to have a very small cross. So we end up with L, L, G, G, and then L, L, G, G. And that would be our cross. Okay, so example 6. This is actually an AP question. And there are 4 points possible, of course. So I want to remind you that you have to answer incomplete statements on AP questions. So I want you to take a moment and pause and read through all of these information that they give you, so that we can do the 2 different parts. Okay, so A says, what conclusions can be drawn from cross 1 and cross 2? Explain how the data supports your conclusion for each cross. So cross 1, the information that they gave us right here, all the offspring had bronze eyes. So the first thing that we know is that bronze is dominant. So down here, cross 1, we're going to say bronze is dominant because all F1 offspring had bronze eyes. So cross 2, what can we determine? Looking at cross 2, we can determine that this is an autosomal trait because there is an even number of male to females in each area. It's very much a 1-to-1 ratio. So autosomal traits. Even number of males and females. Okay, so B. What conclusions can be drawn from the data from cross 3 and explain how the data supports your conclusions? Remember that you can always support with a chi-square value if you need to. Okay, so let's go up here back to cross 3. So we can see the numbers. If you look, we obviously have some linked genes or some recombination going on, some crossing over. So we should have gotten a 1-to-1 to 1-to-1 ratio, and we did not. So we're going to say that linked genes are present because we did not get a 1-to-1 ratio. We also know that crossing over has occurred due to recombinant. And what else can we figure out from cross 3? So explain how the data, so we need to look more at the data here a little bit. So the recombinant, we can actually figure out the percentage of the recombinant and how far apart they are. Because if we add these up, and in the males, for instance, so if we do 220 plus the 260, and we divide that by the total, then we are going to get 0.10, multiply that, it would be 10%. So we can also say that the recombinants are 10 map units apart. Okay. So let's look at some linked gene notation examples. Instead of writing a genotype, big A, big B, little b, linked gene notation can be used to identify the alleles that are found on the same chromosome. So for instance, here, you could have a big A, big B, little a, little b. Okay? So an individual is big A, big B, little a, little b, just like I showed here. So on the following chromosomes, show the genes that would be found on each. So that's what this is. Body color and eye shape are found on the same chromosome in fruit flies. The black body is recessive to the wild type, which is B positive. So for eye shape, lobed eyes, L is dominant to the wild type, L plus. So an individual is heterozygous for both traits, and the wild type genes are on the same chromosome. Write the genotype using linked gene notation. So what this is saying is that we are going to have little b, little l, and then we're also going to have b plus and l plus. So on the chromosome, it would look like this. So the next one says the following parents were crossed. Little b, little l, little b, little l, and little b, little l with b wild, l wild. So determine the expected outcome if no crossing over occurred. So first of all, I just want to note that the wild type, this is just for linked genes. So don't let it confuse you here. Okay, so first of all, we have BL, little b, or little b, little l, little b, little l. So we're just going to use a simplified punnett. And we have a B, L, and a B plus L plus. So the B wild, L wild. So finish this cross. Okay. So when no crossing over occurs, there's going to be that 1-to-1 ratio. Remember that. Okay, so 40% of the offspring were little b, little l. Determine the genotype and frequencies of the other 3 types of offspring that would be born if crossover did occur between the 2 genes. Okay, so we know that we've got 40% here. And this is our expected, our norm, versus our recombinant. So we know that if we have expected 1, over here is 40, then our genotype 2 is also going to be 40. So that tells us that we have 80%, we need to equal 100, so these two would be 10. So the next thing to figure out is what the expected would be. So we know that we have one, we just looked that up here, the BLBL. So the other one is going to be the BL with the B and the L wild. So the recombinant, if crossing over occurred, you took a cross between those, we could have a BL with a BL wild, and we could have a BL with a B wild L. So hopefully you see what I did there. Basically, you've got your BL, BL, and then the other wild types. So basically, just crossing over, so 1 with 1. So calculate the percentage of the progeny that were recombinants. Well, we know here we have 10 and 10, so it would be 20. And then what was the frequency of crossover? Again, 20. Because we have those recombinants. How many map units apart are the genes for body color and eye shape? That would be 20 map units. Okay. So let's review really quick. So on the left, we're going to go through the principles, and then we're going to look at the exceptions. Okay, so we're going to go down the left side, and then we'll come back and go down the right side. So for every trait, each individual has 2 alleles. Each individual passes on only 1 allele of the 2. This is referred to as the law of segregation. There are 2 alleles for every trait, and one is dominant and the other is recessive. There's not a place here for it, but we're going to write anyways. This is called the law of dominance. The inheritance of one trait, such as height, does not influence the inheritance of another trait, such as color. This is the law of independent assortment. And for every trait, 1 allele is from one parent and 1 allele is from the other. Okay, so those are all the principles that you need to know. Now let's talk about the exceptions. Some traits, such as eye color, are polygenic, which means they're determined by more than one gene with 2 alleles. If eye color is determined by 3 different genes, then 6 alleles code for the trait, not 2. Some traits, such as hemophilia, are sex-linked, since the gene is found on the X, and the male only has the Y for the trait. Some traits arise due to non-disjunction, like Down syndrome, where a parent passes on one allele instead of one of their 2. Some traits are going to show multiple alleles, like blood type, and there are 3 alleles for blood type in our population. In addition, A and B are codominant. Some traits, like sickle cell anemia, show codominance, and some traits, like the pea plant flower color, can show incomplete dominance, where neither allele is going to be dominant or recessive. Some traits are linked so that these genes cannot independently assort, and the linked genes are sorted together into the same gametes. Some traits, such as color blindness, are sex-linked, meaning they're only on the X. Therefore, males only get 1 gene for each trait on the X, and that is from their mother. For sex-linked traits on the Y, Males only get one gene for each trait on the Y from their dad. Okay, we're going to go through a couple exceptions problems that you could see, and then we will be done for today. Okay, so sometimes results are given and they don't show a pattern. This can be due to what is called an environmental factor affecting gene expression. And this example could be soil pH of a plant, or it could be maybe the amount of sunlight. It could also be due to stages that an organism is going through. So it could change colors as they mature or develop more hair, etc. It could also be due to another gene affecting the expression of a gene. So in example 1, this flower is often seen in the late summer and early fall. It's an exotic looking plant that is native to southern Brazil and typically is found hanging. The flower is normally red, but sometimes the yellow or pink flower variation is found. The data above were obtained after several crosses. Which of the following statement best explains the data? Okay, so first we've got a red and yellow cross, a red and pink cross, and a pink and yellow cross. So let's see what our options are. So A says the appearance of red in F1 generation of the pink and yellow cross demonstrates that flower color is not an inherited trait but is determined by the environment. Well, they didn't give us any data about the environment, so we're going to say no to that. B, the flower color depends on stages of flower development, and young flowers are yellow, advancing to pink, and then red. Well, they didn't tell us anything about what stages any of the flowers were in, so we're going to get rid of that. C, since the F1 and F2 phenotypes of the pink and yellow cross do not fit the expected genotype and phenotype ratios, the flower must reproduce by vegetated propagation. Okay, this is something that we have never talked about. We will never talk about because it's not something in AP. If there is something that you do not recognize, you have to trust that we have talked about everything that we need to that AP covers. So this we're getting rid of because it's not something that we've ever talked about. And then D, flower color is an inherited trait, and the F1 and F2 phenotypes of the flower arising from the pink and yellow cross can best be explained by another gene product that influences the phenotypic expression. So we're going to go with D. Example 2. A hydrangea plant has blue flowers when grown in acidic soil, but has pink flowers when grown in basic soil. A clone of the pink flowered plant is grown in acidic soil and produces blue flowers. This change in flower color is most likely due to, well, the key thing here is that they're talking about soil types, okay? So, is that related to sexual reproduction? No. Asexual? No. Environmental conditions? Yes. So, that would be our answer, would be C. And that is the end of this lecture. I will see you guys next time. Make sure that you are going over these examples so that you are familiar and you have lots of practice with your Punnett squares. And I will see you guys later.